Good day to everyone joining us, and welcome to today's X Talks webinar. Today's talk is entitled Optimizing Obesity Trials, Integrating Diverse Endpoints to Holistically Assess Safety, Efficacy, and GLP-1 Agonists and Beyond. My name is Sonia Hunt, and it's my pleasure to be your X Talks moderator for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. Now, this chat box is located in the control panel, which is found on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this chat panel. I'd also like to welcome our attendees viewing this presentation from our LinkedIn Live event, and I encourage you to submit questions as well via the comments tab. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for streaming to those who register on xtalks.com. At this point, I'd like to thank Clario, who developed the content for this presentation. Clario is the leading healthcare research and technology company that generates the highest quality clinical evidence for their pharmaceutical, biotech, and medical device partners. They offer comprehensive evidence generation solutions that combine eCOA, cardiac safety, medical imaging, precision motion, and respiratory endpoints. Clario's science, technology, and operational experts have helped deliver over 26,000 trials and contributed to over 800 regulatory approvals in more than 100 countries. For more than 50 years, they have provided deep scientific expertise and the broadest endpoints technologies to help transform lives around the world. Now it's my pleasure to introduce our speakers for today's event. And first, I would like to introduce you to our co-moderator for today's event, and he is Dr. Todd Rudeau. He is the Executive Vice President and Chief Medical Officer at Clario, providing medical scientific leadership across the organization. And there he is. Hi there, Todd. He has nearly 20 years of clinical cardiology and pharmaceutical research experience with a career predominantly focused on drug safety. Next is Dr. Mark Tangowski. He is the Director of Medical and Scientific Affairs for Medical Imaging at Clario. His current responsibilities include clinical development and trials experiences in SLD MASH, Crohn's disease and related complications, rheumatological MSK conditions, and women's health. And next we have Dr. Paul Strump. He is a diabetes and metabolism key opinion leader. There he is. Hello there. And clinical science consultant at Clario. With over 20 years experience in pharmaceutical development, he advises on diabetes, metabolism, and obesity clinical development strategy for Clario. And finally, we have Dr. Fabian Chen. He is the Vice President and Deputy Chief Medical Officer of Cardiology at Clario. Hello there. In addition to providing leadership for Clario's Cardiac Safety Department, he provides expert consultation to clients on scientific and regulatory strategy. Dr. Chen has nearly 20 years of clinical cardiology and pharmaceutical research experience. Now, with the introductions out of the way, it is now my pleasure to pass the mic over to Todd. So, Todd, when you're ready, you may begin.
Great. Well, thanks for the very kind introduction, Sonia. Appreciate that. Good morning and afternoon, everyone. Thanks for joining us. As Sonia said, my name is Todd Rudeau. Very glad to have the opportunity to host this interesting and really quite packed session today about digital endpoint strategies in diabetes and obesity drug development. Over the next 45 minutes or so, we're going to take you through a range of considerations in digital endpoint solutions, highlighting the role that innovative technologies can play when within a successful development program. On your screen here is our agenda. I'll start us off with a brief introduction just to level set us all on the epidemiology and clinical aspects of obesity that we need to all be aware of. I'll also speak to the recent history with GLP-1 receptor agonists as a drug class and some of the development considerations surrounding this. We'll then hear from Dr. Tangowski, who will walk us through some of the innovative imaging modalities that are applicable in this space, with a particular emphasis in understanding how weight loss drugs may affect muscle mass and visceral fat distribution. Dr. Strumpf will then speak to us about the roles and methods for glucose monitoring and the importance of understanding hypoglycemia risks, as well as some of the applicable patient-reported outcome assessments. And then finally, Dr. Chen will address some complexities of running a drug development program in very high-risk cardiovascular populations like this, and discuss some of the mitigation strategies that one could take. So why don't you go forward, Sonia? Let's see, 2 slides probably, and yep, and I'll start us off with this overview. So obesity is defined by a body mass index of 30 or above. BMI is not a perfect metric, by the way. It's simple and quick, though, and it's essentially just weight in kilograms divided by the square of body height in meters. And while BMI is generally accepted as a good indicator of risk, as mentioned, it does have some limitations I'll mention here. There's a risk that BMI will overestimate body fat in athletes or individuals who have a high muscular build, and it can underestimate body fat in older people or those who have lost substantial muscle mass. But with that said, we still have a major public health problem to address, and it's not just an issue with an imperfect metric. In the US alone, 40% of us fit the definition of obesity, and approximately 70% are overweight or obese, defined as a BMI is 25 or greater. And just to be clear, this isn't a US only problem. The obesity prevalence globally is over 16%. This has doubled in the past 30 years and has quadrupled in adolescents. And why is overweight and obesity such a problem medically? Well, there are so many comorbidities or complications that come along with obesity, some of which I've outlined here on the slide. Insulin resistance and the metabolic abnormalities that come along with that, which includes type 2 diabetes, a marked increase in cardiovascular risk, including high blood pressure, cholesterol, heart attack, risk for obstructive sleep apnea, a negative impact on mental health, increase in certain types of liver disease, and even a clear association with some cancer types. The good news, though, is even a modest change in body weight, a 3 to 5% reduction, can lead to measurable improvement in glycemic control, in blood pressure, cholesterol control. So when we think about the treatment options for obesity, there are 3 major pillars. Lifestyle changes, so that includes calorie restriction, increased physical activity, behavioral therapies, so lifestyle restrictions, changes, pharmacotherapy, and then of course surgical intervention as well. In medications, there have been some successes, including the most recent success of GLP-1 receptor agonists, but there have also been some failures. So drugs that are stimulants to increase the basal metabolic rate have adverse cardiovascular complications, for example, or drugs that prevent fat absorption like orlistat, which is mentioned here, have notable gastrointestinal side effects. And while seemingly drastic, perhaps bariatric surgery has actually shown some particularly notable benefits that should be mentioned here, the decision to proceed in that direction, of course, is quite individualized with consideration of benefits and risks. But in some cases, may be a very appropriate decision. So weight loss at 2 years can approach 70% of excess fat, which is unsurprisingly, therefore, accompanied by really remarkable improvements in blood pressure and blood sugar and lipid profile in patients who have this done. You can go to the next slide, please. So today we're mostly hearing news about GLP-1 receptor agonists, and the exciting aspects of this drug class in particular are the fairly remarkable efficacy in combination with high tolerability and a favorable safety profile. And that's, of course, a recipe for success. They are by no means new, however. In fact, originally discovered in the mid-'80s and subsequently developed specifically for type 2 diabetes management. The first approved drug, GLP-1 agonist drug, was approved nearly 20 years ago in 2005. So what's this drug class all about? Well, GLP-1 is a hormone, and it's a hormone that promotes insulin production to control blood sugar after a meal. It also suppresses appetite. So GLP-1 agonist drugs, they mimic the action of that hormone. And therefore, not only do they improve glucose control, but they also slow the movement of food out of the stomach. So the patient feels fuller for longer. And in the development of these drugs for diabetes, it was observed that meaningful weight loss was also occurring, and subsequently development programs were pursued in non-diabetic patients that were overweight. And as you undoubtedly know, we now have drug approvals that are not just in this class for diabetes management, but also approved purely for weight loss in non-diabetics. So you can see here in the graphic on the right a comparison of weight loss magnitude that's observed in different studies of different GLP-1 agonist drugs. One in particular, semaglutide, has been demonstrated to reduce risk for adverse cardiovascular events in non-diabetic patients. And then just a couple of weeks ago, you may have seen there was a phase 3 study with that same drug that demonstrated efficacy in steatohepatitis, which is fatty liver disease. Even more remarkably, this class exerts a seemingly beneficial anti-inflammatory effect. And so we're now seeing a range of indications that this drug is being explored for, mostly in phase 2, neurodegenerative conditions like Alzheimer's and Parkinson's, inflammatory bowel disease, and even mood and cognitive disorders as well. So the last thing I'll mention is an important topic that you'll hear much more about from Dr. Tengowski next, and that's the concept of where that observed weight loss is occurring. Well, obviously our goal is to lose excess body fat, but what we do observe is some loss of lean muscle mass as well. And with respect to losing body fat, there are some nuances there also. So visceral fat, that's fat that sits within the organs or surrounding the organs, is very different than subcutaneous fat, which is the fat that sits just underneath the skin. So visceral fat generally has health consequences that are more significant, and simply stepping on a regular scale at home, you're not able to differentiate what type of weight loss may have occurred. So leveraging different imaging modalities, again, which we'll hear about shortly, can provide insight to this and help a drug developer understand how their drug is working, but also help to differentiate one drug from another potentially. So with that, you can go forward to the next slide. I will hand it over to Dr. Tengowski to talk a bit more about this in detail.
Great, thanks, Todd. I'm turning my camera on. It's great to get an opportunity to talk with everyone today. I'm going to take the concept of sub-Q versus visceral fat just a little bit further, a little bit detailed here, because I want people to be interested in these two fat compartments. We used to think that fat was just fat and that we would lose it everywhere or anywhere. That isn't really the case today. We look at the two fat compartments. The other thing is fat is actually biologically active, and that's important as well. So we know our sub-Q fat lies just under the skin. Its purpose is to pad the muscles and the bones, and it's thickest around the hips and the thighs, around the belly. It's involved in thermal regulation, and it contains a special connective tissue that holds our body together. The downside of that, think of cellulite, that's your connective tissue there. So it aids in the connectivity between the skin and the blood vessels and the nerves. It's really governed by genetics, and there's a lot of different genes that go into fat distribution. On the visceral side, that is the deep fat that surrounds our abdominal organs, around the bowel, for example. It is about 10 to 15% of our total body fat, and it is highly lipolytic, and it's the release of that free fatty acid that travels through the portal circulation that then can pile up in the liver and may be a way of regulating our NASH, the proton density fat fraction that we see in the old NASH studies or the new MASH or SLD studies. That fat contributes to liver steatosis and hepatic insulin resistance. It's common in adults, but I also want to mention that kids today, we're seeing as young as 5 are having real builds up to this fat, and that may be a consequence later on in their life. Visceral fat has some direct effects on the function of the sub-Q fat, and what we're really concerned with are the inflammatory mediators that are produced by the fat, and those enter into the bloodstream. And there's genetics that are linked to your fat expression, IL-1B, and this laminin gene expression. So there is a genetic component, again, to visceral fat. Let's go on to the next one. Okay, so what is the risk benefit? And the risk here is thinking of skeletal muscle versus the benefit of fat. And we know that patients on GLP-1 agonists can lose 25% of their weight rather rapidly, but that muscle loss is not always associated with weight loss. And that's where we have to ask the question, does too much loss of muscle mass present harm in the long term to people who lose a lot of weight? Is the loss of that lean muscle mass making you fatter or fattier if you look at my lean mass versus my fat mass? We'll talk about some methods of measuring that. So what is this long-term effect? What does it have on bone mineral density? We're deloading the frame. Do I need to have big bones or high bone mineral density if I'm not supporting all that body weight? So for many folks, they say, the body image is really important, but if it's at the expense of mobility or general physical function, the question is it really worth it? And how can we have subjects lose weight without introducing some other problem just because they want to weigh less? So we can look at the cardiovascular benefits. We're going to talk a little bit about that later. And then we also have to look at the free fatty mass versus the skeletal muscle mass, and where are we losing weight? Because I don't want subjects to lose weight but lose so much muscle mass that they're actually fattier at the result of their pharmacology. So there are some things that we can't move. We can't move fluids, we can't move organs, can't move body tissues, and we have to look at where from men versus women. We know that men have less fat. They tend to have a greater proportion of weight loss from the free fat mass versus a skeletal muscle mass than women do. So it's an important consideration of both short-term gains in your trials and long-term gains as we think about safety. And I know it's those long-term that are going to be very important as we get more longitudinal data. So let's go on, please, and we'll talk about what kind of weight loss happens. And we can, in the interest of time here, maybe where are we losing the muscle? What is the risk of the losing muscle versus fat? And then what happens to skeletal health? Do we produce a situation where the fracture risk goes up because I don't have muscle mass tugging on the bones, stimulating them to remain dense? So let's go on to the next one and think about what kind of imaging endpoints can you have, as I like to say, below the neck. I'm going to have one slide at the end. I'm going to talk about above the neck. But right now, below the neck, I need to look at adipose tissue. I need to look at muscle. I want tissue fat content, and I have to look at the skeleton. All these collectively give me ideas about what were the drug actions, what was indirect, what's related to nutrition, mechanical unloading. All these things are factoring into your interpretation of both efficacy and safety. And the main modalities that we use for this are DEXA, MRI, and we've done some CT, but mostly due to radiation exposure, we really don't use CT in our trials. So let's get into the detail of what each of these modalities can do for you when developing an obesity trial and efficacy or safety endpoints. Let's please go on ahead. So coming out of the MASH space, getting into obesity, liver fat as assessed by MRI-PDFF is pretty much the universal non-invasive gold standard. I've got, from a physics perspective, I've got protons. They're precessing in space. They're in alignment with the field of the magnet, and I'm pulsing these protons. And whether they're a proton in water or a proton in fraction in a fat molecule, like a free fatty acid, I can exploit those differences because fat precesses ever so slightly faster than water. So I can distinguish, and this is the chemical shift part, fat versus water in my acquisitions. That's the in and out of phase that I get to generate water and water plus fat images. The downside to fat, as you can see, I've got CH3s and CH2s, and I've got a big NMR spectrum. Fat, however, is never in phase with itself. So there's always a little noisiness, if you will, in the fat image, but we're pretty comfortable with what fat versus fat and water mean in our images, and the acquisition fairly standard. I can do this on 1.5 Tesla scanners or a 3T scanner. The in-phase, out-of-phase timing is slightly different, but that's already worked out. We know GE, Siemens, and Philips acquisitions are all pretty much, let's say, universal. I don't have to apply any correction factors to them. And it's actually a fairly easy procedure. There's no contrast given. There's no, it's all done under breath hold. So these aren't long, multi-minute acquisitions. So I'm laying on the bed. I position my coil over my abdomen. I do my scalp planning to make sure that I'm getting the entire liver scanned. From that output, I will generate a fat fraction map, and then I'll draw ROIs in each of the QNO segments, get individual fat because I do see heterogeneity of fat across segments, but it's the overall fat fraction and that fat fraction change over time that really tells me if I'm having an effect on liver fat. So this is a modality that's validated across field strengths. It's great for central QC review and reading. It does require special software, so not everybody can do this, but we're finding that because of the obesity studies, more and more big medical centers do have advanced liver imaging capabilities. One thing that we do have to do, though, is we're finding that some machines, like some Siemens scanners, that's actually some Siemens software, we're seeing a higher than expected rate of fat-water swap in those images, which make it hard for us to make a fat fraction map. So we have to train the techs at the sites to look for this artifact, because in most cases, it's a total swap, not a partial swap, and they're used to seeing partial swaps because the images don't look right. Let's go ahead, please. And now we'll talk about stiffness. Stiffness and fibrosis mean different things. Fibrosis is more of a histology term, but stiffness is what I can do for MR elastography. I have a need for a resoundant acoustical generator. That's the blue and white box there. I put this little driver on the liver, not high enough into the lungs, and not low enough that I'm only getting the posterior right lobes. I want to get 4 slices. I'm putting mechanical waves into the liver, and what I'm doing there is looking for the transduction of sound across the liver. That's what the wave image is showing you. So sound traverses the liver. It also can work on the spleen. If I'm going to interest in spleen stiffness, though, I would reposition the paddle, do 2 acquisitions. Out of this wave image, I generate my elastogram and the confidence map. That's where the readers put their ROIs, and then I get a stiffness scale related to how fast did that sound traverse the liver. Again, this tells me about stiffness, not necessarily fibrosis, but it is a good surrogate for liver stiffness. Anything that we can do to reduce biopsies are really good here. So liver stiffness is a reliable measurement. It's amenable to central review. It does require the special hardware and software. And large subjects who have a big sub-Q fat mass, we can only turn up the amplitude of sound so much, and they can be problematic in imaging. But we do find that at least 1 or 2 of those slices in a 4-slice acquisition, we can get usable ROIs. So let's go ahead. And let's look at MRI for both whole body and sat-vat analysis. So I can do whole body analysis. These are breath hold acquisitions. There's many of them. It is a survey, if you will. It's not a detailed analysis, but I can do a lean fat analysis of skeletal muscle across the abdomen and into the legs. It does measure, I'm going to talk a little bit more about the sub-Q fat that we do in our acquisition, where we look at the spine as the landmark. You'll find some papers will say, use the umbilicus as a landmark. Well, it turns out that if you lose a lot of weight, your umbilicus is in a new location for that follow-up visit. If you lose a lot of weight, chances are your L2, L3 disc space, though, hasn't moved very far. We need a single slice for sat-fat analysis. We will capture multiple slices, and we will generate this sat-vat map, if you will, where we've segmented out all of the fat, and then we've divided up what is a sub-Q, that's purple, what's visceral, that's yellow, and then I can do ratios of those two. Let's go ahead. Ectopic fat here, I just talked about sub-Q fat, but if I think about fat in muscles, I can do this in the shoulder, in the thigh, in the gastroc. I get very high resolution images. I'm using the same fat acquisition techniques, Dixon methods, PDFF, and I look at the anatomic scans that are T1 weighted, and I look at the fat scans from the Dixon and the fat fraction. And from here, I can segment out what's the sub-Q fat, what's the fat between muscle bundles, and then what is within an individual muscle itself, what is the fat content. I can apply this method. It's widely available on other MRI scanners, and I can do the pancreas and the kidney as well if needed. But most of our muscle work, we rely on these techniques with anatomic and fat scans. CT can be used to validate techniques, but it's really radiation says don't use it in a trial. So I think 2 more slides to go here. Please go ahead. Great. So let's talk about body composition by DEXA, because I think this is the bang for your buck method. I can do a whole body scan. I can segment out what is the bone mineral density from the fat. I can look at anatomic regions of the arms, the chest, the android gynoid regions, look at the visceral fat that's been validated against CT measurements. Whole body DEXA, widely available, patients accept it. It's a reasonable, very low radiation dose. Measurements are reproducible, they're accurate and precise, and it does give us a rapid indirect measure of adipose and muscle. So let's go ahead. Thanks. So sorry, my last slide here, fMRI and the gut-brain axis. So functional magnetic resonance imaging, or fMRI, this is a non-invasive diagnostic technique that looks at brain activity as a function of blood flow. So as you put in agonists, as you put in antagonists, you alter that blood flow, and I can detect that through my analysis and say what are the nuclei within the brain that are activated or inhibited based on the way molecules work. So I have, in a typical endocrinology, I have an agonist and an antagonist. So ghrelin is one that stimulates appetite, gastric motility, and food intake, while the GLP-1s actually seek to reverse that effect, and they want the pancreas to stimulate insulin release, suppresses the secretion of glucagon, it slows gastric emptying, and subjects just don't feel hungry when they're on a GLP-1 agonist. So when I look at fMRI, after those injections, I can see where ghrelin has increased the blood flow in these brain nuclei regions, and where GLP-1s decrease flow. So you can ask the question, is my molecule getting into the brain? Is it having a central effect in addition to a peripheral effect? So this is a nice complement to all the outputs from the bone, fat, muscle analysis, is to say, am I stimulating the brain to respond as well to GLP-1 agonists? So that's all I have. I hope there were some questions in there. Keep them, put them in the chat. I'm going to turn the talk over now to Paul, who's going to tell you about glucose. Thanks.
Thank you very much. May I have the next slide, please? So the question is, what is COA and eCOA? Here on this slide, we see 4 different acronyms that are components of COA. Let's define COA first. It stands for Clinical Outcome Assessments, which are used to measure how a patient feels, functions, or survives, and they can be recorded by a patient, clinician, uh, or a caregiver or observer. As seen here, they can also be performance related. Today, we will focus mainly on number one, which is the PRO, or patient-reported outcome assessment. This involves having patients or participants report their own responses. eCOA is simply the electronic version of such data collection. For example, an electronic patient-reported outcome, or ePro, is when you have a participant answer survey questions on a tablet or handheld devices. In GLP-1 trials, electronic patient-reported outcomes are designated in smartphone apps to ask participants questions about their experience, symptoms, and blood glucose meter readings. Next slide, please. Thank you. So what is SMBG and CGM? Self-monitoring of blood glucose, or SMBG, is the measurement of fingerstick capillary blood glucose by a patient using a drop of blood and a lancet device to prick the skin. The blood is then applied to a blood glucose test strip that is inserted into the BGM. The blood sample requirement is quite small, 0.3 to 1 microliter, and modern blood glucose meters, BGMs, have memories to store the blood glucose values and may have features like Bluetooth connectivity to sync data with smartphone apps. A continuous glucose monitor, or CGM, captures real-time interstitial glucose data every 1 to 15 minutes. The interstitial glucose value is mathematically converted to a corresponding capillary blood glucose value. This provides a more comprehensive assessment of glycemic control than a BGM. The granular CGM data allows researchers to evaluate key metrics like the percent time in range, which we call the target range, of 70 to 180 milligrams per deciliter, time above and below this target range, and glycemic variability, which are important indicators of diabetes management and the risk of complications. Next slide, please. Thank you. Low blood glucose, hypoglycemia, can be caused by many medications used to treat diabetes. Insulin and sulfonylureas cause the highest rates of hypoglycemia. In 2023, the FDA released a draft guidance for industry on diabetes about efficacy endpoints for clinical trials in drugs being used to treat diabetes. Within this guidance, FDA noted that hypoglycemia endpoints have primarily been used to evaluate safety and only rarely used as endpoints for comparative claims, i.e. efficacy or comparative safety. SMBG data have been used extensively to populate these hypoglycemia safety endpoints in the past. Next slide. This is about hypoglycemia reporting. Hypoglycemia, as you can see on the left, is characterized as level 1, which is a level sufficiently low for treatment with carbohydrate. Typically, there are symptoms of hypoglycemia which can occur, including palpitations, tremors, hunger, sweating, confusion, difficulty with speech, drowsiness, blurry vision, lack of coordination, nausea, or headache. Notice that there is a blood glucose cutoff for this of less than or equal to 70 milligrams per deciliter. Level 2 is sufficiently low to indicate serious, clinically important hypoglycemia, and there is a blood glucose cutoff here of less than 54 milligrams per deciliter. And level 3, previously called severe hypoglycemia, which has cognitive impairment and requires the assistance for recovery. In clinical trials, case report forms, let's call them reports in this context, can be designed so that one report captures level 3 hypoglycemia and another captures documented hypoglycemia, which is level 1 and level 2. And again, note that level 3 hypoglycemia is not defined by a discrete blood glucose value. Consider the following hypoglycemia minimum reporting requirements at the bottom of the slide to satisfy regulatory safety reporting to support successful approval. This is also an opportunity for a robust patient-reported outcome, PRO framework, to be considered. Some other considerations for reporting include time frame, which should be all-inclusive across the study time frame, e.g. since the last visit, whether there were any symptoms present or not, and we can ask this as yes, no, or unknown. In our experience, the regulatory authorities have not required that the exact symptoms related to hypoglycemia events are included in the event report, just if the event was symptomatic or asymptomatic. Next slide, please. Hypoglycemia does occur, and let's use an example for Ozempic. In this example, from the US package insert for Ozempic, we note at the top of the slide that even in the placebo arm of an Ozempic monotherapy placebo-controlled trial in type 2 diabetes, there is hypoglycemia reported in this low-risk arm. As expected, the risk is higher at the bottom of the slide when Ozempic is added on to basal insulin with or without metformin. In conclusion, there is a need to proactively collect data on hypoglycemia in order to populate the safety database in studies of anti-hyperglycemic drugs. Next slide, please. Blood glucose meter versus continuous glucose monitor in clinical trials. The use in clinical trials of BGM is summarized on the left, and for CGM, it is summarized on the right. BGM represents a point-in-time assessment and is therefore used when symptoms of hypoglycemia occur and to guide treatment and response to treatment if value is less than 70. In summary, BGM data is accepted for documenting hypoglycemia safety data. CGM, on the other hand, collects aggregate glucose data and is ideally suited to comparing different CGM metrics such as time and range, rates of hypoglycemia, especially nocturnal hypoglycemia, which may be asymptomatic between arms of a study. CGM can also compare trends over time. In summary, consensus for use of CGM data to evaluate hypoglycemia is evolving, and discussions with regulatory authorities are appropriate for defining appropriate endpoints. Pragmatic recommendations for monitoring. Next slide, please. It may be appropriate to have different strategies for monitoring of hypoglycemia in type 2 diabetes studies versus type 1 diabetes studies. A pragmatic approach is presented here for type 2 diabetes at the top and type 1 diabetes below it, with noted use of blinded or unblinded CGM. Next slide, please. There are hundreds of validated assessments that can be used to measure patient-reported and clinician-reported outcomes. Just a small sample of some of the most commonly used assessments are shown below. Now, when these assessments are collected electronically, whether they're patient-reported or clinician-reported, we unlock significant advantages: higher quality data, improved accuracy, and consistency in line with regulatory expectations for contemporaneous attributable data collection. Next slide. The data in the paper diary, shown on the left, eventually has to make its way to an electronic format. And you can just imagine the challenges for translating and transcribing paper into electronic databases. That is compared to the efficient transfer of electronic data that is automated and error-free, as shown by the electronic diary on the right. Next slide. We are not alone in this assessment. There is widespread regulatory support and guidance to use electronic data capture for clinical outcomes assessments, as shown here. Next slide. These 7 reasons for using electronic data capture for COAs are not exhaustive, but here we highlight some high-level advantages of electronic data capture. 1, the high quality data that meets regulatory guidelines around eCOA standards, and critically for at-home data capture that we collect contemporaneous data. 2 and 3, having real-time insights to patient safety and study performance allows for pre-built alerts and reporting capabilities, and a centralized approach to tracking both safety and operational metrics. 4 and 5 are highlighting the advantages around shorter study timelines as well as costs. These benefits over paper are rooted in operational efficiencies during study timelines as well as cost. The study itself and the elimination of timely processes for handling paper data collection, entry, and storage is an advantage. Finally, critical to diabetes trials in particular, note the importance of patient engagement and compliance and the ability to integrate with glucose monitors. Next slide, please. Initial research is signaling a potential benefit to mental health from GLP-1 receptor agonists. Over to the left, you'll see a systemic review summarizing potential impacts, both positive and adverse, on mental health outcomes. Ongoing research is examining this area closely, especially in light of recent concerns about associations between GLP-1 receptor agonists and suicidal ideation or behavior. So far, evidence does not support a strong link. Still, continuous monitoring is critical, especially among patients with existing mental health conditions, to ensure everyone's safety. Next slide. It is vital that any thoughts or behaviors related to suicide are carefully monitored. One gold standard for this purpose is the Columbia Suicide Severity and Rating Scale, or CSSRS. Relative to the paper version, electronic data collection of the CSSRS offers key advantages, as was previously discussed. Clario, in partnership with Dr. Posner, has migrated this assessment to an electronic format. The electronic format allows for immediate reporting and programming checks that notify sites and sponsors in real time of any positive findings. Over on the right, you see Clario's patient-reported version. Notably, this method encourages higher sensitivity in capturing patient data compared to the clinician-led interviews, as it supports greater candor and minimizes rater bias and variability. Next slide. Thank you. On this last slide, let's focus on 4 key considerations for gathering patient-reported outcomes, or PROs, in diabetes trials. First, ensure that collection of safety data meets regulatory agency expectations in diabetes trials. That means integrating blood glucose monitoring data that capture and report hypoglycemic events accurately. A daily electronic diary designed to track hypoglycemia can streamline this process. Second, having a well-rounded ePro strategy is vital. This involves setting compliance expectations, programming period, recall options, and implementing branching logic and reminders. Equally important is training participants not only on the device, but on the relevance of the questions so they can provide accurate, meaningful responses. Participant training is also vital to protocols that integrate glucose monitor devices alongside electronic diaries. Lastly, real-time reporting tools and electronic PRO collection systems enhance oversight. This capability is especially valuable for monitoring hypoglycemic events and participant compliance with daily entries, both critical to maintaining participant safety and data integrity throughout the trial. Thank you, and I'd like to hand this presentation over to Dr. Fabian Chen.
Thank you, Paul. Great. Next slide, please. The results from several large cardiovascular outcome trials, some listed below, demonstrate that GLP-1 agonists provide a cardiovascular benefit. For example, the LEADER trial for liraglutide. And as a result, organizations such as the American Diabetes Association and the European Association for Study of Diabetes recommend GLP-1 agonists for type 2 diabetic patients with high cardiovascular risk in order for them to reduce adverse cardiovascular outcomes. Taking this a step further, the FDA has approved indications for semaglutide that also include improving cardiovascular outcomes in patients with obesity who do not have diabetes, in addition to lowering blood sugar in type 2 diabetic patients and chronic weight management in adults with obesity or who are overweight. Next slide, please. How do GLP-1 agonists improve cardiovascular health? As my colleague Todd Rudo spoke about in the beginning, GLP-1 agonists can improve cardiovascular outcomes by reducing appetite and inflammation. They can decrease adiposity, decrease body weight, decrease hepatic steatosis or fatty liver, decrease lipids such as LDL cholesterol. Additionally, GLP-1 receptors, which the GLP-1 agonists bind to, are expressed in the heart and vasculature. This suggests that there could be a potential direct effect of these drugs on these tissues. Evidence supporting an improvement of cardiovascular health by GLP-1 agonists include preclinical studies, which demonstrate, for example, that these agents can reduce atherosclerotic plaque, There are non-randomized clinical studies that demonstrate that GLP-1 agonists can improve heart function, reduce the infarct size in patients with myocardial infarction, and reduce coronary artery inflammation. And again, as I mentioned in the previous slide, there are randomized clinical studies that demonstrate that GLP-1 agonists reduce MI, cardiovascular death, and stroke events. Next slide, please. So how do we optimize clinical trials that involve GLP-1 agonists and similar agents? Participants in GLP-1 agonist and obesity trials often may have higher cardiovascular risk. Thus, examination of cardiovascular benefits in these patients who have higher cardiovascular risk requires use of both cardiac safety and efficacy endpoints. The next few slides will cover these endpoints, and they're listed below: ECG arrhythmia, blood pressure, cardiac blood biomarkers, cardiac imaging, patient and clinician-reported outcomes, which you just heard about from Paul, which I will cover, and also clinical cardiovascular endpoints. Next slide, please. ECG and arrhythmia. The traditional ECG shown in the figure in the bottom left is a mainstay for assessing cardiac safety across all indications. This can be performed both in the office and at home, shown just right of the traditional ECG is a home ECG device that we also support. However, many cardiac toxicities cannot be assessed well with the 12-lead ECG, and this is because these cardiac adverse events are less frequent and occur outside the office. Examples include heart block, bradycardia, atrial fibrillation, ventricular arrhythmias, and these are better assessed by Holter or patch monitoring devices shown on the right bottom. And these can be used to monitor patients for extended periods of time outside the office. Next slide. Blood pressure assessment. Some studies have demonstrated that GLP-1 agonists can lower blood pressure, but this remains to be determined still. However, blood pressure, similar to ECG, is a mainstay for assessing cardiac safety across many different indications. This can be performed in the office or at home, and can be performed while the patient is ambulatory. And devices for this are shown below on the slide. Next slide. Cardiac blood biomarkers. They can be used to assess the potential for GLP-1 agonists for improving heart failure. BNP, brain natriuretic peptide, and the N-terminal pro-BNP are both very useful heart failure biomarkers that are used clinically, but can be used in trials to diagnose and to follow patients for improvement or worsening heart failure. Since many patients in these trials are at higher cardiovascular risk with the potential for myocardial infarction or cardiac injury during the trials, myocardial injury biomarkers such as cardiac troponin I or cardiac troponin C, which are commonly used clinically to take care of patients, can also be used to follow these patients in trials to diagnose and assess for myocardial infarction and injury. The advantage of blood biomarkers is that they are relatively inexpensive, they're easily repeatable, they can detect cardiac toxicity well before imaging studies can be used for the same purpose, and they also may allow prevention of permanent ventricular damage by giving the investigator time to intervene. Next slide, please. Next slide. Cardiac imaging assessment. To follow on Mark's discussion, for cardiac imaging, this includes echocardiogram, computed tomography or CT scan, magnetic resonance imaging, and nuclear imaging, which could include PET, SPECT, and MUGA, which I won't get into. These imaging modalities are very useful to diagnose and characterize a variety of cardiac adverse events, including heart failure, left ventricular dysfunction, myocarditis, and pericarditis, for example. Next slide. The next slide is an example of echocardiography. And shown on the right is an image which is in motion of Parasternal low axis view of the heart. Also noted above is pericardial effusion and pericardial fat. Shown on the left is a cartoon of a patient with visceral obesity, and a cartoon in the middle shows the parasternal axis of the heart with pericardial fat shown in yellow around the heart. Next slide, please. Last, cardiovascular clinical endpoints are often the basis for regulatory approval for cardiovascular indications. And composite clinical endpoints often used in cardiovascular trials include mortality, which could include all-cause death or just cardiovascular death, major morbidity, which includes typically non-fatal myocardial infarction and non-fatal stroke. And for heart failure trials, this includes cardiovascular death and heart failure hospitalization. Most often, the cardiovascular clinical endpoints in a trial are managed by an independent endpoint adjudication committee. And the benefits of this is that these committees ensure accuracy, reliability, and impartiality, especially when adjudicating endpoints. This improves the credibility of the trial data and reduces the perception of bias. And this can be very useful when the endpoints are complex and subjective. This is the end of my section. I'd like to turn it over to Todd.
Great, thanks, Fabian. Well, sincere thank you to all of the panelists for their contributions today. It's quite a session there, dense with considerations for digital endpoint strategies in diabetes and obesity drug development programs. You've heard about the role for different imaging modalities to further understand the type of weight loss absorbed. Dr. Strumpf reviewed the importance of hypoglycemia assessment, not just in measuring blood glucose, but also understanding the concurrent symptoms. We heard about the criticality of robust suicidality monitoring and other patient-reported outcomes. And then we just closed there with Dr. Chen's review of cardiovascular safety monitoring strategies. And I think the key takeaway there is understanding the importance of sufficient baseline testing to establish pretreatment risks and then appropriate on-treatment monitoring and event adjudication to detect a safety signal. You can go to the next slide. So with that summary, we're going to transition to a brief Q&A segment in the remaining time, which I'll host here. I'll invite the panelists now to turn on their videos so they can rejoin us. We have a good number of questions that have come in already during the session, so thanks for your engagement there. Please continue, though, to use the chat box to enter any questions or the comments field if you're viewing on LinkedIn Live, and I think we'll get started. I will say, though, if there's any questions we don't get to today, you can look out for some directed email follow-up. We'll be sure to get back to you individually. This first question here, directed to you, Dr. Strumpf, looks like you're going to predict the future here for a second. What does the future regulatory landscape look like for the evolving inclusion of continuous glucose monitoring in efficacy and safety endpoints?
Thank you. Well, of course, we don't have a crystal ball, so can't definitively comment on future regulatory policies. My personal thoughts I can share, and that's that we see this field evolving, and FDA has evolved to consider CGM data to populate efficacy endpoints, as I pointed out in the guidance earlier in the presentation. And it is encouraging that there is regulatory recognition that CGM systems have advantages over SMBG, specifically to assess hypoglycemia as an efficacy endpoint, and that also, the timing of when the hypoglycemia occurs may favor CGM, such as nocturnal hypoglycemia. Given that the use of CGM systems in the regulatory context is evolving and various uncertainties that exist to using this technology in clinical trials, the guidance itself recommends and encourages sponsors to engage with the agency regarding the use of CGM and CGM-based hypoglycemia endpoints. So conversations with the agency are encouraged, and the agency states that position in the actual guidance. And this is all good news for this important evolving endpoint.
Okay, great. Appreciate that, Paul. Thanks. Good question there. Second question, this one's going to get to an imaging question. So for you, Mark, you presented several different modalities to assess body composition, including DEXA, CT, and MR. Can you speak to how long it takes for a subject to get these different types of scans, and any other considerations when comparing one to another? That's a good question now.
Yeah, so the DEXA scan is a fairly rapid scan. Depending on what so you're looking at, 10 to 15 minutes for a DEXA scan. For the liver scans by MRI, you're looking at 20 minutes. Because you got between the scouts, the anatomic scans, the advanced liver package imaging, non-contrast breath hold, there's time that's required for reconstruction. You're looking at about 20 minutes of scanner room time. If you add in a SAT-VAT, now I've got to reposition my subject. That's probably going to add another 10 minutes to my protocol. So the longest one I would say is the whole body imaging, or the more detailed skeletal muscle. The more I add in, some of those methods can be in the 40 to 50 minutes collectively. None of these scans are really long, but when you layer one upon another, it's not so much a burden to the subject, other than just having to lay there and the magnets banging away, but it's more about the time and the cost that goes with occupying the scanner room for that length of time.
Okay, great, thanks. Let's do one here for Dr. Chen. That's interesting. It's about QT monitoring. So do you need to assess for pro-arrhythmic cardiac safety risk in trials of new obesity drugs? Kind of a broad question there.
Thanks, Todd. Yes, especially if the new molecule is a small molecule drug versus an antibody or a protein. And the pro-arrhythmic risk can be assessed through a thorough QT study. That's typically often done later in development. However, one could also use a concentration QT analysis during phase 1. And this is something that we at Clario are happy to help with.
Okay, thanks. And maybe we can squeeze one more in here before we need to wrap. Dr. Strumpf, this one's going to go back to you. Can you share some more with us about designing a hypoglycemia diary, and any guidance regarding what symptoms should or should not be included in that diary?
Yeah, thank you. We have to be very sensitive to burden, both on the study sites and the patients in the trial. So there has been success that I'm aware of listing specific typical symptoms of hypoglycemia in the diary and in the protocol, and asking very simple question when you have a documented hypoglycemia event, that's a blood glucose value less than 70 by SMBG, or if you're using CGM endpoints, similar cutoff, is to ask, were typical symptoms of hypoglycemia present, and offer the responses of yes, no. And then, of course, if it's not answered, then in the case report form, you can also have a choice of unknown. This strategy has served well in phase 3 studies that I'm aware of that have led to approval of therapies for diabetes.
Great, thanks. All right, and apologies, I know there are a few more questions here we didn't get to. We will follow up by email. Sonia, I will hand over to you to close us out.
Okay, well, thank you very much, Todd, Paul, Fabian, and Mark, for that great presentation and discussion and for answering all those questions. And just like Todd mentioned earlier, if we could not address your questions, the team at Clario will follow up with you after this presentation. Okay, and now, before I share that email address, if you have any further questions, we do have something here to mention. For those of you interested in hearing more from Clario, meet up with Mark, who will be attending the AASLD 75th Liver Meeting to learn more about the Designing Holistic Obesity Trials with Multidisciplinary Endpoints. And click on the QR code here to get that. And I'm also going to put the link in here. Mark, you want to tell us the day and time?
Yeah,
A little more
thanks
info there?
everyone. Most of us have already got our reservations in. The liver meeting this year in San Diego begins on Friday, goes through Tuesday. I will be there the whole time. Please contact me via email or LinkedIn. You'll see me hanging around the exhibit hall. I tend to walk around there as well as the poster sessions. You can either contact me or just bump into me, and yeah, I would really enjoy the opportunity to tell you a little bit more about imaging and fatty liver and obesity trials.
Okay, thank you very much for that, Mark. Thank you everyone for participating in today's webinar. You will be receiving a follow-up email from X Talks with access to the archive for this event, a recorded archive for this event. A survey window will be popping up on your screen. Your participation is appreciated as it will help us to improve on our webinars. Additionally, I will be sharing a link to view the recording of today's event in the chat box, which you can also share with your colleagues so they may register for the recording here as well. So give me a few seconds to do that. And for those of you watching via LinkedIn Live events, we encourage you to visit xtalks.com to register for this event and gain access to features such as viewing, the recording, and more. Now, please join us in thanking our speakers for their time here today. So that is Fabian, Paul, and Todd, especially Todd for being my co-moderator, and Mark. Thank you, Mark, for letting us know about the AASLD 75th Liver Meeting. And here is the email address you see here on your screen. If you have any further questions, please utilize this email address, science@clario.com. We hope you found this webinar informative. It has been my pleasure to be your webinar moderator. On behalf of the team here at X Talks, we thank you for joining us. I'm Sonia Hunt. Until next time, please take care and bye for now. Bye, everyone. Bye, Todd, Paul, Fabian, Mark.
Bye.
Bye,
Thank you.
everyone.
Bye.
Bye.